Good morning and good evening, everyone. Welcome to the Consortium of Communitarian Universities of State of Rio Grande do Sul, COMUNC, webinar series in Brazil. My name is Professor José Miranda, and I'm the head of International Office of La Salle University Canoas in Brazil. I will moderate this webinar. It's a pleasure to receive the audience for another webinar promoted by the Internationalization of Higher Education group in COMUNC, where the best researchers from each university present their work. The Consortium of Communitarian Universities of Rio Grande do Sul State is formed by 14 universities with more than 8,000 professors and more than 11,000 employees with almost 200,000 students in a universe of around 450,000 students enrolled in the state of Rio Grande do Sul in the south of Brazil. It offers around 1,400 undergraduates and graduate programs, making a suitable and reliable higher education system in operation in the state of Rio Grande do Sul. Community universities as COMUNC are not public, as the Brazilian government does not maintain them. However, they are a public asset, a through network with education, science, and technology, covering almost all municipalities in the state. They are an authentic public, non-state, non-profit, democratic, and participatory universities that prioritize quality in teaching, research, and extension. Considering the internationalization process in which these higher education institutions have been involved, many challenges are encountered. It's an honor to receive in this webinar series Dr. Gustavo Fioravante Vieira from La Salle University in Canoas. He is graduate in biological science with emphasis on molecular, cellular, and functional at the Federal University of State of Rio Grande do Sul. He has a PhD in science by the graduate program in genetics and molecular biology of the Federal State of Rio Grande do Sul. He is currently professor at the postgraduate program in health and human development at La Salle University, and also a collaborator professor at the postgraduate program in genetics and molecular biology at the Federal State of Rio Grande do Sul. He has experience in the field of immunology, with emphasis on understanding the structural mechanisms that govern the immunogenicity of T cells for application in the development of rationalized vaccines. He founded and coordinates the Nucleus of Immunoinformatics at the Department of Genetics at the Federal University of State of Rio Grande do Sul. Also, his work in this area. He received also a grant in the Bill and Melinda Gates Foundation in 2009 for the Grand Challenges Exploration Program, round second. It has collaborations with other structural bioinformatics projects involving a study of oxytocin variants in the study of the alpha-I hydrodanase enzyme, coagulation factors, and HIV resistance to drugs. Dr. Gustavo Vieira will bring us a conference about his research at La Salle University. This title of presentation is Bioinformatics in COVID-19 Vaccine Development. Any question regarding the conference can be made using the chat. At the end, Professor Gustavo Vieira conference will pass the questions to be answered. Professor Gustavo Vieira, welcome, and please, you can begin your conference.
Good morning, everyone. First of all, I would like to thank Professor José Miranda for his kind introduction. So I will present my work entitled Bioinformatics and COVID-19 Vaccine Development. My name is Gustavo Fioravante Vieira, and this work was made in collaboration between Universidade Salle and postgraduate program in genetics and molecular biology of Federal University of Rio Grande do Sul. So COVID-19 brings us some scenes of high sci-fi movies to real life. When we could imagine that more than 40 million people could be infected now for a totally new virus. Here at this website of Johns Hopkins University, we can see that more than 1 million people died for this disease. And here we can also see that the top infected countries are composed by US, India, Brazil, Russia, Argentina, Spain, Colombia, France, and so on. So while this number is extremely high, we should consider that 40 million people is less than 1% of the whole population, world population. And considering the power of dispersion of this virus, this number could be even higher. So this is denominated the iceberg effect. The iceberg effect take in consideration a huge number of individuals that are not diagnosed, are not recognized as infected individuals of COVID-19. This group of individuals are composed by individuals possessing COVID-like symptoms in anamnesis without any hospital admission and diagnostic tests, individuals presenting no symptoms, although close contact to COVID-19 patients, and asymptomatic virus carriers. And the size of this group of individuals is still unknown. But the iceberg effect is caused by underreporting errors or prior immunity. This immunity or this immunogenicity elements could also influence differential outcomes in infected populations. So in this presentation, I'll try to elucidate some elements that could be influence these answers, okay? But first of all, what is immunogenicity? Immunogenicity is the ability of a particular substance, such as antigen or epitope, to provoke an immune response. So, deciphering the elements that trigger defenses of immune response is the big riddle. I choose this image to be in the background of the Oedipus and the Sphinx that illustrates this paradigm, this conundrum. Okay, deciphering the elements that trigger defense of immune response is the big riddle. When we decipher these elements, we could improve the health methods the approach to avoid many infectious disease by the understanding of these mechanisms. So to start our specific issues, our specific points, we should start with a basic immune toolbox. So when a virus infects an individual, two main arms of defense are enlisted in our bodies. On the left, we can see the humoral immune response, and on the right, the cellular immune response. The humoral immune response is the response responsible to produce antibodies, and these antibodies will bind to the viral particles, neutralizing these virus to avoid that they infect the cells. And this task is performed by this specialized kind of cell, the B lymphocytes. So when we talk about antibodies, we are talking about these molecules here, these Y-shaped molecules, and not this bunch of cells. These cells are named immune cells, not antibodies. Antibodies is just these molecules that binds to the viral particles. And many studies, many discussions around the world now about COVID is focusing in the production and the generation of specific neutralizing antibodies. But there is another arm, very important in this process, the cellular immune response. This cellular immune response is performed by two kinds of cells. The T helper cells that will secrete cytokines here, and these cytokines works like signals to inform the B lymphocyte and the T lymphocyte that that environment is dangerous. And these specific cells on the right, the cytotoxic T cells, will investigate cells to check if they are infected by a virus or bacteria or becoming tumor cells. And this investigation is made by this receptor here in the cell membrane, named T cell receptor. So this T cell receptor will interact with the MHC presenter molecule in the cell surface of nucleated cells. And this molecule will present small fragments of proteins named peptides. These peptides could be derived from self or from non-self proteins. When the peptides are derived from self proteins, the recognition does not occur, but when the cell are presenting non-self peptides, the T cell lymphocyte is able to recognize that the origin of this peptide is non-self, and then they eliminate these viral factors because the virus invades the cell to replicate and to generate new viral particles. So our study will focus this interaction between the T cell receptor and the MHC present peptides. The peptides are small fragments of the proteins. The proteins are the sections or parts of a whole particle, a whole cell. The cells, the virus, and the bacteria are formed by proteins. Proteins, and the proteins are formed by amino acids, and little stretch of amino acids are named peptides. So proteins in the cytoplasm will be degraded by the proteasome, and this proteasome will pick up the protein, generating peptides, and these peptides will be translocated by this specialized protein named TAP, and these peptides inside the endoplasmic reticulum can bind or not the MHC. So some peptides have the ability to bind, and others don't possess this ability. And when a peptide binds to the MHC, it will be exported and presented to the TCR, that is the T cell receptor of cytotoxic T cell that we are seeing here. And this process is denominated MHC1 antigen processing pathway, which is the main route to permanently check the intracellular peptidome content. So one more time, I bring attention to this interaction. This interaction will define if this cell, this specific cell, will be eliminated or not by these specialized immune cells. And this elimination will be dependent of the recognition or not of this small fragment of proteins. This mechanism allows that these cells could check if this internal environment is infected or not. But how it occurs? When we see this interaction will be depicted in the next slide. Here, we can see the TCR in blue and purple, this TCR, now in three-dimensional structure. And the MHC is depicted in orange and pink. The MHC is this molecule, and the peptide is represented by the red spot inside the cleft of the MHC. So here, another representation showing the TCR, remember the TCR, the MHC presenting the peptide, and this scanning, this interaction will define if this cell will be eliminated or not. So our existence as a human depend on this correct recognition all the time in each cell of our bodies. So we will investigate how this recognition or not occurs by this slide. Here we can see the point of view of the TCR, the T cell receptor, how the immune cells see these peptides inside the MHC. Here we can see the MHC con the peptide in the cleft. This peptide is formed by atoms. These atoms possess charges, positive and negative ones. And this whole set of atoms in fact generate a molecular surface, and this surface has a electrostatic potential distribution. So the T cell receptor see the topography and the charge distribution of these molecules. We can see the blue dots representing positive charge, the red dots negative charge, and the gray zones neutral charge. So it's important to understand a pivotal point here. TCRs, T cell receptor doesn't read. T cell receptor doesn't read. It's pretty obvious, but it's very important to consider because approaches just comparing amino acid sequence by alignment process are not so meaningful. But why? For two reasons. First, peptides with a high identity can produce very different molecular surfaces. And two peptides possessing very divergent sequence can generate similar surface. And for the point of view of the TCR, what really matters is similar surfaces. This QR, remember, it seems a QR code, immunological QR code. This distribution will inform the TCR if some cells is infected or not. This is the fingerprint that informs the T cell that some is infected or not. Well, in the context of COVID-19, we analyze these targets by the point of view of the sequence versus structure, investigating cross-protection in the COVID-19 context. What we did was to recover. We have a previous infection by a SARS virus in 2003, causing the SARS in that occasion, with a reduced number of individuals infected. But in that time, some peptides, some stretches of proteins were identified as immunogenic. So we recovered these sequences by this database denominated Immune Epitope Database, which is a public repository. This database contains epitopes or peptides, immunogenic peptides from any virus, from bacteria, from tumor cells. And these peptides were experimentally described, experimentally obtained, and deposited in this database in a curated way. So we could recover specific epitopes, specific peptides for the coronavirus. Here we choose the coronavirus using the host, the humans, only with T-cell assays, and these assays only for positive one. That is, we only recover a sequence possessing recognized immunogenicity and involved in infectious disease. The next step, with this step, we recover 20 sequence, what we can see here, the 20 sequence, each one of them represented by a sequence of amino acid. And these sequences are derived from different sources. For example, the spike protein, nucleoprotein, all the proteins of coronavirus, SARS-CoV-1, the SARS-CoV involved in the 2003 epidemics, nucleoprotein, and membrane glycoprotein. If we have this sequence involved in the trigger of immunogenicity in a highly related virus, we could compare this sequence with the current strain, the current virus. So we recover the corresponding sequence in SARS-CoV-2, the virus that causes COVID-19. And as we can see, 15 out of 20 peptides present the identical sequence. Blank cells means no amino acid difference, so this sequence is identical to this sequence. And in this case, the only difference is the substitution of a leucine for a serine and a threonine by serine. So the mutations that usually occurs among the viruses are represented in bold. But when we consider that the TCR only recognizing a morphological change, not the sequence of letters, remember, TCR doesn't read. So we should investigate this alteration in a structural way. Here, we also recover corresponding sequence from other coronavirus. This coronavirus 229E, NL63, and OC43 are coronavirus that are commonly involved in common flu, common cold. So here we can see some difference probably in comparison of sequence methods. We could neglect this sequence because they don't present a high identity, conspicuous similarity, okay? So, thinking that regard to vaccine development, we could use identical or highly similar peptides, already described as immunogenic in SARS-CoV-1. So, if we know that a specific sequence in SARS-CoV-1 was immunogenic, and this sequence is the same in SARS-CoV-2, we could use this sequence in a immunogenic method, like a vaccine, for example. We could include this sequence because if it is identical to the former sequence, probably it will be immunogenic in the current virus. Nevertheless, it does not explain previous high immunity, considering the low number of infected individuals on that occasion. We should remember that in that occasion, a small number of individuals were infected by the SARS-CoV-1, and it probably is not conferring immunity to the whole population in the world, a huge number of individuals that do not present severe symptoms in SARS-CoV-2. So another virus probably is giving this previous immunity. We should remember that previous infection is like a vaccine. When you is infected by a virus closely related to a new one, you probably is protected against the second one. So here, you take a look in these two peptides from SARS-CoV-1 and SARS-CoV-2. Here we can see only a substitution of a valine by a methionine. And here we model this peptide in the context of the MHC. And here we can see the point of view of the TCR to conclude that probably the former virus strain could be conferring protection or could be used as an immunogen in vaccines approach working for SARS-CoV-2. And how we obtain these structures? We have a method named Doctop, a tool developed by our group. And this method allows that we can input a sequence of peptide, and this sequence will be modeled inside a specific or desired MHC. And after this structure is obtained, we can calculate the electrostatic potential distribution around the molecular surface, and we could analyze, now compare these peptides in a structural way, not only comparing the sequence. So here we can conclude that these surfaces are very similar, so this is a good candidate to vaccine approaches. But here we can see a very interesting example. This peptide from influenza virus, when we compare with these two sequences, are very different, very contrasting. So in a regular comparison among sequence, this candidate probably would not include it. But here, using a method of structural modeling, we can see that this peptide from influenza virus, the virus flu-causing virus, this peptide is even more similar to the SARS-CoV-2 than the peptide for SARS-CoV-1. What is the significance of this comparison? Here we can see that probably individuals that were infected by influenza virus could have an immunity against the SARS-CoV-2 disease. So some individuals that have influenza, that have a flu, could already be protected against SARS-CoV-2. This could give us some protection to avoid a more disseminated pandemics. And here we compare our peptides from SARS-CoV-2 against a panel of immunogenic peptides contained in this database, named Crosstope Structural Data Bank for Cross-Activity Assessment. This database was established by our group, and this database possesses a panel of immunogenic peptides modeled in the MHCs, and it works as a panel of sketches to compare immunogenic fingerprints with our candidates to vaccine development. So when we compare peptides from SARS-CoV-2 virus against this panel, we find some interesting similarities. Here we can see a peptide from SARS-CoV-2 similar to vaccinia virus. Here we can see a peptide from SARS-CoV-2 ALN-TLV KKL, similar to a peptide from hepatitis C virus. And here we can see a peptide from SARS-CoV-2, similar, the peptide is very different, as we can see, LA-LL, DRL, similar to human herpes virus 4. And what we can conclude here is peptides from SARS-CoV-2 can be similar to peptides from individuals from 3 different viral families, as poxviridae, flaviviridae, and herpes viridae. virus that regularly infect humans. So this is a very important, pivotal information that previous infections along the evolution, the human evolution, could be protecting us against new emergent viruses. And here we also can see that SARS-CoV-1 peptides and other coronavirus peptides that causes common cold are also very similar. So we can, both coronavirus or these representatives of the different viral families can give us some protection against SARS-CoV-2. Another point considered by us was the HLA. The HLA is the MHC in humans. The MHC is the presenter molecule that we see before. HLA, genetic diversity, and differential response to COVID-19. We know that the MHC is the protein codified by genes localized in the most polymorphic area of the human genome. So these polymorphisms are concentrated in the MHC cleft. So these mutations allow that different MHC could present a different set of peptides. This variability allowed that the human population could respond against a myriad of different viruses. So only in humans, only for MHC class 1 MHC genes, or one specific class of MHC, we have more than 7,000 different proteins codified by humans. So this variation was selected by the evolution. Here we can see that individuals, homozygotes, are negatively selected after different stressing conditions like war, intensifying competition, scarce resources, poor nutrition, and increasingly poor health. Or, for example, in episodes like the current episode, as a result of pandemics. So usually homozygote individuals for this protein are selected, being the heterozygote positively selected. So there is a trend to select diversity in these proteins in the human population, allowing that the human population construct a shield against future pandemics. And here we can see, for example, genes, the frequency of genes for the HLA-B genes, the MHC frequency of specific HLAs in different countries. Here we can see, for example, that the B57 possess 7% of the individuals in Canada has this allele, 5% in Northwest Russia, 0% in Japan, 3% in Thailand, and so on. And another alleles, there is more discrepant frequency as B53, 27 in Barbados, 5% in Brazil, 42 in Cote d'Ivoire, and 0% in Thailand and Japan, for example. So there is a variation of the number of individuals possessing different types of the HLA around the world, and probably this will give a differential ability to this population to deal with each specific virus. So we investigated the involvement of this variation in the context of COVID-19. In this case, we recover frequencies from the most affected and the countries possessing the high rates of fatality and the low rates of fatality during these pandemics. And we recovered alleles in high frequency in these two conditions. And we calculated the weighted average for each one of the alleles recovered. We performed this recovery frequency from the allele frequency net database. This public database contains information regarding frequency of different alleles in different populations. And start from this frequency, we performed a hierarchical clustering analysis. This kind of approach allowed that we could cluster similar individuals here, for example, individuals wearing similar styles, or women and men in different groups, only putting some features of these individuals. In our case, we only input the frequency of each allele for each specific country. So just inputting this frequency, we could obtain different clusters. Here I'll only show one cluster format, and we can cluster countries like Spain, Italy, and France in one cluster, Germany and Russia in separate clusters, China, Iran in another one, and Brazil, US, and UK. But what is special here? We can see that Brazil, US, and UK present similar death rates, the same for Spain, Italy, and France, high rate of deaths, and Germany and Russia present low rates of deaths. But this clusterization do not take in consideration any information regarding the number of individuals that were infected, or that were deceased, that death in after the disease. The only information to form these groups were the HLA frequencies. So this is indicative that the pool of HLA present in each specific population could give differential ability to deal with this specific disease like COVID. Probably another virus like HIV or other emergent virus could generate different clusters because the pool of HLA will be different to fight another infection. So only imputing HLA frequency from the different population, we were able to cluster countries with similar death rates. And so the concluding remarks in our study were HLA genetic constitution among affected countries could partially explain differential degrees of severity and fatality observed. So both sides are important. The side of the population where the genetic constitution could give some differential ability to fight against the virus, and heterologous immunity triggered by previous infections by viruses commonly faced by humans could be acting like a force field, reducing the number of deaths and severe cases. So probably a fraction of the population already possess a hurdle effect, a previous defense against the current virus, because the current virus has stretches of proteins similar to stretches of proteins from influenza or herpes virus or hepatitis C virus. So individuals that already have some of this infection, but not all the individuals, this is very important to stress. Some individuals that generate a response against influenza could be generating a response against a specific stretch of the influenza virus that is similar to the sequence in SARS-CoV-2 virus. Many individuals that already have influenza infection could not be protected. So it's very important to maintain the orientations to avoid the dispersion. So thank you very much. We know that we have a long way to walk, and we are just in the beginning. Thank you. Professor Miranda está mudo.
Perfect. Thanks, Professor Gustavo, for your wonderful explanation. Certainly, we are facing difficult times right now, but also many research like you are doing right now will bring us important benefits for the future. Just a question I have for you, considering your work and what you are observing all around the world in terms of research. How is Brazilian right now in terms of research in your area? We can consider that we are doing well comparing to the other countries?
I think that the main difference it refers to the budget.
Yeah.
This is very important in science. For example, U.S. invested $10 billion in the vaccine development. So in Brazil, we should to invest in creativity. So my field of research is immunology. Usually, immunology is performed by experimental assays. But for to perform a specific assay, we should spend around $100,000 or even more only to start the experiments. So it's probably impossible in the current status of our research. So my group invested in bioinformatics analysis or computation analysis in the last 15 years because we can perform studies that were published in highly relevant journals, like journals from the Nature Group and PNAS and Journal of Virology, because we invested in creativity in cheap experiments, only using a computer and internet-based tools, because in terms of experimental to be applied in individuals, we don't have a budget necessary to spend these experiments.
Perfect. We got a question for Fabi Sartori. He congratulates your studies and asks, being able to identify different frequencies, clusters, you can also identify specific populations' abilities to fight against the virus?
Thank you for the question, Fabiani.
Fabi Sartori.
Fabi. Thank you for your question. It's very interesting. And the expected step to infer more precise inferences should be genotype the individuals that had the COVID-19. When we genotype in the whole number of individuals that was infected, we could infer more specific response regarding the real power of the genotyping, the allele frequency in their ability to fight the disease. For example, when I know that individuals that have a severe phenotype was from A type of the allele, and individuals that don't have the severe or mild condition or asymptomatic individuals are from C genotype, we can differentiate in a more appropriate way which were the alleles involved in the disease progression and so on. So in our case, we just give a panel to give the first clues demonstrated the involvement of HLA and the disease severity. But to extract more information, we need a wide-scale genotype of these alleles in infected individuals.
Thank you, Professor. In the name of the Consortium of Communitarian University of the State of Rio Grande do Sul, I would like to thank you, Dr. Gustavo Fioravante Vieira, from La Salle University in Canoas for his participation on this webinar series. I also would like to invite the audience for the next Comung webinar series in the 3rd November with the name Migration and Refugee, Actions Adopted by the Faculty of Law of the University of Passo Fundo with Professor Michele Chiuco. Thanks for your attention and see you soon in our next Común webinar series. Bye-bye and thank you.
Thank you all.